Thank you for the invitation and the ability to share our data on our pandemic program. My talk will go into basically our platform for our Vero Cell derived vaccine, and then cover all the work that we did prior to the H5N1 pandemic with regard to our H5N1 program, and then follow with what our response was to the H1N1 outbreak. So first, for those of you who don't know, our vaccine is manufactured in Vero cells, not embryonic hen's eggs. This has a great advantage in that Vero cells work well on microcarrier beads. That's what you see up here on the left-hand picture, top panel. This is Vero cells on an electron micrograph of Vero cells on a microcarrier bead prior to infection, and then on the right panel, the cytopathic effects of high replicating virus on the Vero cells. We use wild-type viruses for our H5N1 program in Europe, not attenuated reverse genetic reassortants. This, as I will mention later, has a huge advantage in terms of speed. We use whole virus vaccine. It's non-adjuvanted, and it's manufactured in our GMP-licensed BSL-3 facility. So what you have in the middle panel is actually the top half of our 6,000-liter fermenters that span two floors in our Bohumil facility in the Czech Republic. We get high yields from our fermenters, 2.2 million doses of the H5N1 vaccine per fermenter, with an overall annual yield estimated to be 100 to 120 million doses. On top of that, this platform can be used to generate split, subunit, and live attenuated vaccines. Vero cells are widely accepted by many regulatory authorities and have been the basis for manufactured and licensed products in more than 60 countries. For more than 30 years, it has been used to manufacture polio and rabies in many countries. Baxter's cell line is a fully characterized continuous cell line, and it supports US and European licensures for our vaccines. It's a serum-free, protein-free medium. It's free of antibiotics and egg proteins, and we get high productivity, not just for influenza, but many other viruses in Vero Cell. Probably the greatest advantage with respect to pandemic vaccines is that it has faster production time than the standard conventional egg-based influenza manufacture. So here you have it. On the top, you can see the first 3 bullet points under the egg are what really caused the delay. For us, as soon as we've secured the viral strain, we can inoculate cells and initiate our manufacturing process. With regard to egg, you need to secure the egg supply, create reassortants, and then test the viral reassortants for safety prior to moving into manufacture. We estimate that this is faster by between 8 and 16 weeks. Could be critical in a vicious pandemic. So just to let you know a little bit about the work, the extensive work that's gone into our H5N1 program, we've done 7 studies to support EU licensure, which includes greater than 5,500 subjects who have been vaccinated. I'm going to take this time to discuss our dose escalation study, our booster study with Indonesia, and then our phase 3 Mockup study in adults and elderly. Just to be clear, our program is based on the A Vietnam 1203 2004 strain with interspersings with Indonesia. So this is our first dose escalation study that we did with this product. It evaluated 4 different dose levels, 3.75 to 30 micrograms in 6 different formulations. This was with and without adjuvant. Aluminum hydroxide adjuvant was the adjuvant that was used. And there were 2 dose levels, 7.5 and 15, that did not use adjuvant. Subjects were vaccinated on day 0 and day 21, and what you see here are the immune responses at the various time points. We evaluated, so it's a little hard here, but obviously you can see that pretty much everyone was seronegative at baseline. And after one vaccination had robust responses across all age groups, clearly not the target response that we would like for protection, but still there was a response both from 3.75 up to 30. What we quickly discerned, though, was that with the groups without adjuvant actually had a higher response than groups with adjuvant. And so we narrowed down our assessment to look at 7.5 versus the 15 microgram. And indeed, there was no, while both were robust, there was no benefit to the added 7.5 micrograms. So there was no added benefit with the 15 micrograms, so we selected the 7.5 microgram for future development. In the interest of looking at what cross protection we might get, we assayed the sera for distant clades, so Hong Kong 156 and Indonesia 05. And you can see that we got strong responses for these cross-clade strains. And we had persistence out to day 180. We then followed this with a booster study. We really wanted to mimic what would happen if we were to evaluate if a group were to be primed with one vaccine and then the strain that came along was something completely different, how would our population go? So we took the subjects who had been enrolled in the phase 1-2 dose ranging study and asked them to participate in a booster study 12 to 17 months later with A Indonesia. So again, these are subjects that were primed with A Vietnam and then boosted with A Indonesia. The total number of subjects was 77 out of the 141. And you'll see that the numbers aren't great, ends of 15, 17, 12, but it really doesn't matter because all doses responded strongly, as you can see, to, I'm sorry, this pointer is not very good, but if you look at the green bars, those represent the A Indonesia that was boosted. So in all cases, we got a strong response to that. If we look at the light blue bars, that's to the Vietnam. So again, we got a strong response there. And then to the distant Turkey and Hanoi strains, we also got responses. I think this suggests to us that this is a strong candidate for a prime and cross-clade boost strategy. Finally, we conducted a phase 3 mock-up study to confirm the dose that we had selected, the 7.5 micrograms, in both healthy adults and elderly subjects. Again, subjects were vaccinated twice at 0 and 21 with the A Vietnam. And again, you see strong responses after one dose, but still not meeting the European criteria for protection. And that subjects, but that there was a lower baseline titer. But certainly after 2 doses at day 42, we see responses both in healthy adults and the elderly that met licensable criterion. So we completed that trial and submitted it to the agency, and in March of 2009, we received our license for the H5N1 program. No less than 2 months later, the swine flu outbreak was initiated, and we got our first, we requested the strain from CDC. We received it in-house and did our first infection in our 6,000-liter fermenters in May. We got our first batch out of that fermenter, actually we got 3 batches out in July, and by August we were shipping it. At the same time we started a study in elderly and adults, and then followed that with children to confirm the safety and immunogenicity of this H1 product. And just so, and I will go into those first two here. We also did a post-licensure study in 9,000 subjects. So here are the immune responses for the H5N1. I think it's important to point out what others have commented on, that these, there was some serious baseline prevalence here. As you can see, 49 and 45%, 36 and 34 in the younger age groups. And in fact, after one vaccination, the responses do not look like our H5N1 program at all, but much more like a seasonal response. And after 2 vaccinations, while we did see an improvement, the improvement was negligible compared to what we would have seen with an H5 vaccine. We also went into the pediatric population, and I think we were just discussing some of the safety observed with, in peds. This is broken out. These are the responses in infants 6 to 35 months in the red bars up here, and I apologize for the mixing of the two. But down here we have 3 to 8-year-olds in the red and 9 to 17-year-olds in the blue. Overall, I think the takeaway is that the vaccine was quite safe. All adverse events reported were of low frequency, the most common being fever in the young infants that lasted less than 24 hours, and that was approximately 13.5%. If we look at their responses, I think this is also an interesting takeaway slide. If we look over here, we have the 9 to 17-year-olds who look very similar to healthy adults, as was previously mentioned. Baseline titers are existent, and after one vaccination, the responses are quite strong. If we look in contrast to the 6 to 35-month-old subjects, clearly this population is naive, and after one vaccination, there is not what we would consider sufficient protection. But after 2, we do get up there. So, and then this 3 to 8-year-old population is somewhere in between, kind of what you might expect. So I think we were fortunate to have our vaccine included in an effectiveness study that was conducted by iMove and funded by the ECDC in Europe and coordinated by a stellar group called Efficacy. This was conducted on the small island of Jersey, which is a British Crown dependency, 118 square kilometers. It has a population of 91,000. It's served by a central health and social services department. It has 1 public hospital and 96 general practitioners. Sort of a captive population, if you will. And in this study, this is an independent study. They tracked influenza-like illness cases, swabbed those cases, and then analyzed the swabs by real-time PCR or culture. One of the benefits of having this confined space was that the distribution of vaccines was quite well tracked. So healthcare workers received our vaccine, Salvapan, and as did pregnant and postpartum women. Children 6 months of age to 9 years of age received Salvapan, and children 9 to 18 Pandemrix. And then students returning from the UK and poultry and pig workers also received our vaccine. There was substantial and rapid uptake of the vaccine in nearly all populations. I think the weakest here is the pregnant women, and the children 5 to 9 years old had a great uptake. There were, of the swabs that were collected, there were 261 cases from the pediatric population starting on week 45 of 2009 onward. among these, the estimate for vaccine efficacy was 100%. That is, there were no cases in this population where the subjects had received a single vaccination 14 days prior, versus 21 cases in the controls. That is, those people who did not have an injection or did not have it 14 days prior. I think it's pretty strong efficacy, but when the confidence interval was calculated, it came out to be 70 to 100%. When it was taken, it was the group wanted to determine exactly how many cases were offset by this protection. And so using the confidence intervals, if the vaccine was 100% effective, we decreased all the cases under this curve, under the red, and if it was 70% effective, that would be the yellow. What was calculated was it's really 132 to 244 cases were prevented by vaccination. There are a couple of caveats in there. One is we don't know what sort of disease amelioration came from that. There's clear evidence that vaccination not only prevents disease, but also reduces the severity and duration of illness. The other is that clearly herd immunity probably played a role in the overall protection observed, and that's not taken into account. So this most likely underestimates the total protection, but it's something that we can fix our hat on. And then another question that we had is this really, is this reduction that we are observing a real reduction as a result of intervention, or is it maybe just the natural logarithm or pathogenesis of the pandemic? And so we compared Jersey's outcome to what was observed in England. And what you see in yellow are the Jersey H5N1 cases, and here in yellow are the English H5N1 cases. Clearly, there was a decrease in cases observed from week 48 onwards in Jersey. I think that's really telling, which is right here. And I'm sorry, I can't hold it so well, but if you look at week 48 in the UK, there's still a huge amount of cases that are occurring. Well, we can't do any cause and effect with these data. I think they clearly suggest that the transmission was stopped by something, most like some exogenous factor. And the fact that the vaccination coincides with the decrease of the illness and distribution suggests that it was vaccination that caused the curtailment of the outbreak. So in conclusion, I want to say I think it's clear that a lot of work was done with our pandemic program. We've looked at, I think, H5N1 and now H5N1 6 ways till Sunday, and we are able to elicit a safe and well-tolerated response, and the vaccine itself is highly immunogenic, with a high percentage of subjects in all age groups demonstrating seroprotective levels. I think the other important factor to consider is that this, our prime boost strategy is well supported by our data, which seems that if we prime, it doesn't seem to matter which priming strain we use. If we follow up with a cross-clade strain, we get strong cross-clade protection. And then, of course, the data from Jersey that I just showed you suggests also that the vaccine was effective in a pandemic setting. And finally, our Vero cells are made quickly. Our Vero cell vaccines are made rapidly and more rapidly than our standard egg competitors, and therefore may be better suited for a rapid response during a pandemic. Okay. Questions? And I'm sorry, I would be remiss if I didn't thank all of the people who have had a huge impact on this work. Okay.
Thank you very much. I would like to start out going back to the manufacturing technology, and one of the reasons why you are much quicker is because you're growing a wild-type virus in cell culture. Now, I was wondering, will you also see differences in performance, because these viruses are obviously not adapted to growing in Vero cells when you get them? Will there be differences between influenza strains that you have at hand?
Certainly, sorry. Yeah, certainly we see strain-to-strain variation in terms of yield. So if that answers your question.
So, but have you then seen that there are viruses that you cannot grow? I mean, because as Teresa was basically pointing out, and we have experienced this in our process with the FDA, when you are a novel vaccine manufacturer, you have a lot to learn, right? Because the viruses or the vaccine changes every year, and the behavior of the proteins or the viruses changes every year. So what have you done to kind of persuade the regulatory authorities in Europe to convince them that this is indeed a technology that is very generally applicable? In other words, that you can always grow the virus that needs to be grown.
Well, I can't speak to what we've done. I mean, we've clearly delivered. This vaccine has been, the Vero cells with influenza have been played with at Baxter since the 90s. So they have every year not only played with the strains that are available, but then other strains. And certainly I know that there have been a couple of strains of note that were low yielders, but by changing slightly the various components, they were able to increase the yield. Okay. So I'm not the manufacturing person, I apologize, but.
Okay, yeah. Any questions from the audience?
It's on antibiotic. Nice talk and nice data. I was wondering if you could expand a little bit on your y-axes and what exactly you mean by sero.
Oh yes, sorry. Yeah, I buzzed her. Oh, sorry.
And also what you didn't cover are the GMTs. So what kind of GMTs are you getting, and what are you using in terms of your, I thought I saw 1 to 20, if I can see correctly.
Yep. Yep, yes, and I'm sorry, we do this so often. So yes, what is presented is microneutralization titers, especially for the H5N1. We know that avian hemagglutinins do not respond as well to the HAIA or AI, depending on who you ask, assay as they do to microneutralization assays. And so we use microneuts as well as SRH to support our licenses. And what I haven't shown you is the SRH, which is comparable. Otherwise, we would not be able to do it. In our hands, we think that the most predictive of protection is the microneuts. And that's what we report. So, but as I said, the SRH, which is also recognized by the EU authorities, is comparable. And then with regard to GMTs, I did not show them, but they're strong. I don't know. I can't give you the individual numbers, but they're good. Okay.
So I noticed that in your dose finding study, you decided to choose the 7.5 versus 3.
Mm-hmm. Mm-hmm. Yep, very good point. I think the consideration that was made at the time was in a pandemic setting, you need a dose-sparing dose. And so while as you mentioned, at day 180, their drop-off is not as great in the 15 micrograms as it is in the 7.5. There were two considerations. One is that you want to have as little as needed and still get the response you want. And then at day 180, is the circulating strain still present? The consideration was, while it did drop off, probably the H5N1 pandemic had already, or some pandemic had already passed, potentially. So, but the main one is dose sparing.
Any other questions? I'd like to ask one final then follow-up question. In Europe, the product was approved and was also used, and then during the pandemic, EMA started changing their recommendation, because initially they recommended 2 doses for individuals, and then a few months in the pandemic they changed the recommendation for I believe the Novartis vaccine and the GSK vaccine, but I never saw a changed recommendation for Baxter. Can you comment on why that happened? So, in other words, your vaccine continues to be recommended 2 doses for the population.
Yeah, and you've got it quite right. Ours is not an adjuvanted vaccine, and we did not receive the single dose recommendation.
Well,
thank you.
Then I'd like to thank Sarah very much for her nice presentation, and I would like to,